Thanks for joining for this month's session on AI for bioimaging data management. For those I don't know, my name is Ken Shopman. I'm ABRF's Executive Director. I'm going to go over a few updates and housekeeping reminders, and then we'll turn it over to our panel for today. Again, we're going to use the chat if you have questions. It's also a great way to meet each other, indicate where you're connecting from, your institution, your time zone maybe. As I said, post your questions in the chat. The speakers will address those after the presentations. And then if you have suggestions for future programs for this group or other ABRF groups, please send those along to us at abrf@abrf.org. So some updates or future events, including ABRF's new strategy, which we launched at the annual meeting this year, which is built on these 4 pillars. Each pillar has a task force that's working on the specific goals and objectives related to these initiatives. So you'll learn more about their plans coming up very soon. And if you want to get involved in working on these groups, please let us know. This is just a different version with some more details about some of the objectives that they're planning to roll out. The first of which will be sort of a data warehouse activity where we want to build on the success of the recent ABRF compensation survey to learn more and gather more information about the scale and scope of shared research resources, how many there are, how many people work in them, the different technology areas, and so forth. So that'll be an effort that we'll be launching early next year. These are the upcoming events over the next few months, some of which are online, including the continuation of this series. And then for those of you in the Midwest, we hope you'll be joining us next week at their annual meeting in Madison with the famous Halloween costume contest. So you still have time to get that organized for next week. And just an overview of the chapter events, most of which have taken place. They've all been terrific. Fantastic events hosted by our regional chapters, and including, as I mentioned, and here's a quick QR code to join the meeting. They're also going to be announcing, I don't want to disclose too much because it's being finalized. They've been very fortunate to secure a speaker on the new FAIR model, which is a new approach to allocating indirect costs. Kelvin Drogemeyer, a former government official who's been the leader of that group, is being scheduled to present, and there's going to be a virtual option to participate in that session. So stay tuned. The MWACD group is finalizing those arrangements, and we'll be announcing that for all ABRF members to participate in that special session during the MWACD meeting. Again, for those of you who were in our meeting this year in Las Vegas or didn't have a chance to attend, you can access a terrific range of content, recordings, slide presentations, even some AI-generated written summaries of the sessions, all at this link that you can see here on the screen. And then finally, I hope you're connecting in the core community. It's a great way to ask questions of your colleagues and peers on broad topics, specific topics, share updates from your cores about different services and resources that you might have. But more importantly, just to help get answers to questions, chances are someone else in the ABRF community has faced a similar circumstance and can provide you some help. And then I hope you're planning on joining us in Pittsburgh next year at the end of March, a little different schedule. The meeting will run from Saturday to Tuesday, March 28 to 31. Registration will open October 21st, and the earliest rates will be available through the end of this year, including some details that we'll talk about a little bit more detail soon about this session and the mini workshop that'll be hosted by this group at the upcoming annual meeting. So with that, I'm going to stop that share and turn it over to Rich Cole, who will be introducing our speakers.
Okay, before we get to the speakers, just some housekeeping and some upcoming events. So next month, which would be November, because of the holiday, we're actually going to move it one day later in the week. It will actually be on the 12th, which is a Wednesday, and that is going to be the second part of image processing. Now, I bring that up to say that at the upcoming ABRF meeting in Pittsburgh, we're going to be running the AI workshop again that we ran last year, because only a fraction of the people that applied for it actually could take it. But a little heads up is something that we learned from last year is we are going to require of all of those who register for it a quiz, and the quiz is going to be based on topics and content from the 2 image processing ABC workshops or webinars that we've held, which are going to be available on the ABRF website, and Ken will send an email with the links to those. So with also in terms of questions, as Ken says, is you can put them in the chat. Also at the end of this, we'll have time where we'll open it up, and if there's questions in the chat, we'll answer those, and then you'll also have the opportunity just to raise your hand and ask more questions. Now we're going to take the month of December off, and we're going to come back in January with segmentation with Fiji. And those are all the announcements that I have. And without any further ado, I'm going to turn it over to our first speaker, which is Jen. So Jen, take it away.
Thanks, Rich. All right. Can everybody see my screen okay? Cool. Hi, everybody. My name is Jen Tay. And as of about 10 days ago, I am now the former bioimage analysis group leader at the University of Colorado in Boulder. I will actually be moving over to the Van Andel Institute in a couple of weeks. So the topic of the discussion today is going to be about managing microscopy data. Just a little motivation. As we all know, microscopy data sets are growing in size and complexity. We have all these really amazing advanced microscopes now that can capture images in 3 dimensions with time lapse, multi-channel, and in a very high throughput fashion. And so, as these data sets grow, we really have to think about what are best practices in order to manage our data, in order to try to make sure that we can adhere to the so-called FAIR principles, right? I'm sure most of you are familiar with these principles. The acronym just stands for having data that's findable, accessible to as many people and different operating systems as possible, interoperable, reusable. And then I like to kind of throw in, as an image analyst, right? I can throw in one last R in there, which is like making sure that the data analysis side, it's also reproducible. So for my talk, it's going to be a little bit opinionated. I'm going to just share some of the data management strategies that we employed at the University of Colorado in Boulder. So just to kind of give you kind of the scope of the size of the core, in a typical year, we work with about 63 different labs, about 170 different users, and we do both microscopy and image analysis. And we have about 9 different microscopes and 2 analysis workstations. So to kind of give you, the size is kind of like a small/medium-sized core. All right, so this is kind of the outline of my talk. I'll just share some tips about data storage in the core, some personal data management tips, and then I'll talk a little bit at the end there about how, more specifically for image analysis, how I kind of manage some of the code and process data. So this is a typical data pipeline, at least for us. All the images that we collect are digital, so they all collected on a computer. And then there's really 2 ways that the data can go. For users who are affiliated with the Biofrontiers Institute, they go to a managed data server. And then eventually then the user can then copy off the data, or it gets sent to me for analysis. For people who are unfortunately not affiliated with the Institute, that data transfer is a little bit more annoying because then they'll have to bring in their own hard drives to copy the data off the microscope computers. And then, I think this is probably similar for a lot of core facilities, but as a core facility, we do not manage any user data, and this is something that we make very clear, especially to new users. And essentially the reason for that is that all the computers are obviously shared by the users of the microscope. You need these computers, and they need to have hard drive space in order for people to acquire the next set of experimental data. And so what we really do is we always encourage people to plan and book additional time to transfer the data out immediately after each imaging session, right? We do have warning signs everywhere. We tell people that, if somebody's trying to acquire, experiment and there isn't enough space on the hard disk, we could potentially delete their data. And, we tell them that we don't back up our managed data, although, on the back end, we actually do, because we don't want angry users, right? So, and for the most part, I think this kind of policy has worked really well for us. We haven't really had we haven't really had too many issues with like people not taking the data off the microscope. And so then in terms of the data transfer, as I kind of mentioned, at the Biofronters Institute, they have a dedicated data server that's connected via the intranet. So everything goes pretty quickly. And for people who are outside the university network, you can kind of access that using the VPN as well. These data servers are, they're quite complex, and they kind of are a fairly, there's a financial cost to it, right? So they are backed up both internally and externally, and they're also handled by a team of like 3 dedicated IT staff. So the nice thing about having this in-house is that affiliate researchers can basically request an account, and then the storage costs become fairly cheap, right? So they are maybe like 10 cents a gigabyte, and they're all basically subsidized by overhead fees. But of course, this is one of the things that I always talk to whenever there's like a PIR lab that I'm working with, I always ask them to think about the long-term storage as well. I'm sure we're all familiar that the NIH has changed some of their data management policies, and now we have to store out our raw data for a lot longer. And sometimes people don't think about budgeting that into like grant overheads and things like that. But those fees do add up over 10 years, 20 years, right? And like I was saying, like everybody else, unfortunately, doesn't who are not affiliated, unfortunately, don't get to use this dedicated data server, and so everybody else has to bring their own external hard drives. And so, in terms of data management, we don't enforce these rules, but I do have some recommendations that I typically provide to users of the facility. So the following slides, I'll kind of just throw out some tips. And again, like this is somewhat opinionated, right? This is just the kind of like the recommendations I've developed over many years working in the core facility. Although if anybody has any suggestions or things they want to discuss, I'm happy to talk about this at the end of the presentations today. So the biggest thing here is I always recommend that users store their raw data in the native format. And by that I mean like the ND2 if you're using a Nikon microscope, or OIF, CZI, depending on what kind of like microscope you're using. And the reason why I always recommend that people store the files in the native file format is that these files tend to preserve the metadata. And I'll talk a little bit more about what this metadata is, but I think it's really important for us to be able to have the information, again, in order to make the data analysis later on reproducible. One of the other things, and I think Doug Cromie kind of pointed this out, is that these original formats are proprietary, which also means that they cannot be written to by other software. And actually having that kind of property is kind of also helpful in kind of preserving the provenance of the data. So that if you have like this raw native file format, that it probably hasn't been tinkered and or modified by by different software. Of course, like not every computer or not every system supports this kind of native file formats. So I often suggest to users, if they really have to store it in a different type of file format, to use an uncompressed TIFF. And so this is just a picture that I've included in here. So the reason why I suggest uncompressed TIFF is that it is basically just the raw data written as an image file. But if you're using a different file format, like a JPEG, for example, or a GIF, just be aware that those file formats natively include some sort of compression, which may not be visible, but if you really compress it really highly, you can see these kind of artifacts occurring, right? And any kind of data compression involves some sort of data loss or degradation of the data. So I always recommend that people kind of use an uncompressed TIFF. And there are some community standards being developed right now. The OME TIFF is one of them. That's being developed to also include some of the metadata from those native file formats. So speaking about metadata, there's two types of metadata that I like to remind people about. Obviously, the first is just acquisition details. Like I was saying, these are the details that are typically going to be included in that native file format. So this can range from basically what optical configuration you have, like what filters you have on there, what is the time between frames, the exposure time, what objectives you're using, and all these other details that can be kind of painful to write down maybe in a lab notebook. All of that will get stored if you keep the data in the original file format. The other thing that I think sometimes, especially I think sometimes people forget to note down with these movies or these images is that you really also should be including your experiment's details, right? So what is the type of cell that you have in these pictures? What are the genetic modifications you've made? What is the passage number? How many generations has it been? Who is the person doing the work? Because as we all know, biology sometimes can be a little bit like a black art, right? Different people can just have different results for no reason. And then I think more importantly, actually also recording in what are the pre-culture conditions, right? So how was the sample grown? Was it grown overnight? Was it like an old sample that was sitting out on your bench for a couple days? I think all those things need to be recorded in order to really make this work reproducible, right? And then in terms of being able to store and manage all that data, what I typically recommend for our users is just to use the native features of your operating system, which is essentially like just using folders and your file structure and file names in order to organize the data. I know that there are more and more alternatives out there that help, like software out there that help you to manage your data, such as like Omero. But the reason why I still like having, this just using like the native folders and file structures is that it just removes one level of dependency. So you don't have to have this external software to help with, and that I think helps with like interoperability, right? So if you transfer the data to like a different computer, different operating system, you don't have to worry about having like this software on there. So the key here, I think, if you want to use this method, is that you really should have a consistent file structure and using a meaningful file name in order to make the files findable in the future. So let me kind of just give you an example here. So this is the file format that I've been using for many years, and as it turns out, it turns out to be like documented as like something called the tier protocol. So essentially for every project, any kind of imaging project I work on, I'll have like one top imaging project folder, which I will try to name with kind of like the date of the project, and then basically like a brief description, imaging cyanobacteria or something like that. And then there's a number of files that I typically have in there. So I typically try to have like some sort of README file, which is like, either a Word document or a text file that kind of basically just describes what is the experiment. Maybe kind of a list of like, here's the data that you might find in here. I'm trying to do more of this now. I haven't really done this in the past, but I'm trying to do more of this where as I complete some of my analysis, I try to generate like a report that summarizes like the main findings in that particular project. And then of course, the most important things in here is these data folders. So I'll typically have a data folder where I'll store, and then subfolders in there where I'll store like both the raw data and then the process data as well. And then I have again, like because I'm an image analyst, I'll have like a scripts folder as well where I store kind of like the scripts for analyzing the data. But I'll talk a little bit more about the scripts in a bit here. So in terms of like just the data folder, again, like I think the key here is to try to use descriptive file names. And so for image files, again, this is just my recommendation. Is I always suggest that people start with the date, but specifically in year, month, day. And the reason for that is if you have year, month, day, if you sort your files alphabetically, these will then sort also chronologically for you. I always recommend that people put the initials again of the person who ran the experiment, and then some sort of link to a reference some sort of link to like a database or a notebook page that contains details. Again, like this is bringing in the experimental metadata that links like basically the experiment to this image file. So, for the most part, there's different ways of doing this, but for the most part, I don't type out the experiments that I run. I don't type it out again and store it in this data structure. I mean, you can, but the way I typically refer to the experimental conditions is that basically we'll have a link to some sort of notebook. So again, like this is just the way I've done it, but basically I'll have something like N2P53, which just stands for, notebook 2, page 53. And then on that notebook page, I'm obviously recording all the experimental conditions that I've done to like get the cells ready for this imaging run. I know as well these days, like the electronic lab notebooks are becoming more popular. So here you might need a different approach. So this is where now maybe instead of doing it with just a simple code, you might maybe you might need to have like a Excel table or something like that where you can have a specific code for this experiment and a link to the electronic lab notebook page. So that was for sorry. So this format that I have here is basically just like what I would suggest for like a single image file. But of course these days we're doing a lot more high throughput experiments, and so kind of like the same thing. If we do high throughput experiments, what I recommend that people do is just store it create like a folder with basically the same information, but then name each individual file depending on what the imaging experiment is, right? So for example, if you are acquiring different XY locations on a slide or 96 well plate or something like that, you could name it the different image files based on that location. And again, all of this kind of helps to make things findable so that now we can use the operating system, like the find feature, stuff like that, to be able to find this data set. And just at a glance, we can also just figure out like exactly what that file is, right? So it makes it a lot easier to locate the data. And I was saying this earlier, so I personally don't do this, but maybe it's something I should do. But the way I've kind of described it right now, where you put like a notebook reference, does mean that you need both the electronic file and you need to make sure you have access to the notebook. It does make the data set a little bit more inaccessible. But if you do want to make it more accessible, you could have a Word document in here that has and kind of like a description of what the experiment, right? But obviously this requires more work to maintain. So kind of just depends. I feel like most users don't really want to do that. So yeah, this is why we haven't really recommended this particular approach. But it's up to you what you prefer. Okay. And then the other thing, of course, is the data backups is incredibly critical. And what we recommend here is that users and labs should set their own policies for data backups. Again, remembering that as a core facility, we don't really handle the data. But what I always recommend for the lab PIs that I work with is that they should really try to think about keeping a permanent record just as you do with a lab notebook. And so I've recommended that lab PIs buy their users, their researchers in the lab like a hard drive, with the expectation that this hard drive stays in the lab just like a lab notebook. And for the users, I recommend that they kind of treat this as like a pristine copy, right? So they really shouldn't be touching these files. And if they want to do any kind of analysis, they should just make a copy of that file on their own local computer, right? So that way, in case anything accidentally gets deleted or modified, you still have all the raw data sitting there. And this is the other thing I also recommend, that users keep their own backups as well, because as we all know, eventually researchers will move on from the labs that they're training in. So I always recommend that users kind of keep their own backups as well. And this can either be in the physical hard drive or on a cloud drive. So it kind of just depends on what they want to do or how much they want to spend on it. And then just the last thing I'm just going to talk about is just a little bit about data management for like an analysis workflow. So for image analysis, really the approach I use kind of depends a little bit depending on like the tool we're using. So I've kind of broken this up into two categories. So one is like a graphical tool, which is like, ImageJ, Fiji, or Maris, or like a programming tool, which is where you're writing actual code, like in Python, MATLAB, or some of the cell profile things as well. So for graphical tools, I'll be honest with you, this is my least favorite to work in because the reproducibility is challenging. You really have to keep like an audit trail when you're working with these tools, which means that you really have to document every action, every button that you click, every setting that you enter. To ensure reproducibility, you really should be recording all these things. The way I do this again is I just kind of keep this with my lab notebook, and that way, there's only like one source of truth. And then I think this is also pretty common advice for everybody, right? But make sure that you record the version of the software and the toolboxes you're using as well. And the reason for that is that, as software versions advance and people make changes to it, sometimes that can affect the internal algorithms and it could affect the raw data value. So you really do want to make sure that you keep a record of this, of all of these different versions that you're using as well. For programming tools, it's a lot easier, I think, because you can save the script. And what I typically recommend for users there is that they should be using some version of version control. Git is I think the most popular version control software out there these days. So this is the software that I typically teach people to use. And the nice thing about using Git is that you can kind of keep track of changes in a particular file, right? So I always recommend that people don't do the thing where they go analysis script version 1, analysis script version 2, and don't have like multiple copies of the same script, but instead just have a single analysis script and then just rely on Git to track the changes as the file, changes over time. And then again, like for backup and sharing purposes, I always recommend that people upload things to like something like GitHub or GitLab. Yeah, I kind of had a little note in there just to make sure that people understand that actually Git and GitHub are not quite the same thing. GitHub is now owned by Microsoft. It's a cloud service that does host your repository, but Git is like the local version of the software that runs and does the actual version control. So I do have some tips on here. I don't think I'm going to go through it in just the interest of time, but like if anybody's interested to talk about this, I'm happy to like discuss it more too at the end of the presentations today. The one thing I will just highlight on here is that if you are going to be using Git or GitHub especially, I highly recommend using this Git tags to track feature changes. And so if you're familiar with Git, this is what basically what a tag might look like. So you basically will specify some sort of version number, and then the way I specify version numbers is to use what's called semantic versioning. This particular format has basically stores version numbers with three different numbers. So there's the major number, there's the minor number, and the patch. And basically you increment these numbers depending on what you're changing in the code, right? So for example, if you're making a change in the code that makes it now incompatible with all previous versions, that's when you want to increment the major number. If you're adding like functionality, so maybe, I don't know, maybe you're adding in a new measurement or something like that to your code, but it's still backwards compatible with all the previous versions, then you would increment the minor version number. And then for smaller things than that, like little bug fixes, things like that, then you might increment the patch number. And then just like the data, I also have folders and subfolders for analyzing for my data analysis. I tend to split this up into two different folders. I tend to have like a processing script. So the processing script, basically you run it on the raw image data, and then it typically spits out, so for example, things like the segmentation masks or the time series data from tracking. And then the analysis scripts are the ones that actually take in this data and generates like the final plots that you might want to look at. So I typically do it this way, partly because that's also the way I structure my code, because typically the processing takes a long time and we'll just kind of like let it run overnight. But analysis, I might want to see it straight away, so I try to split those two functions up anyway. The most important thing when you have any kind of code is, of course, don't forget to write documentation. And this is something, again, like I always teach my students and my users, you're going to make sure that you write documentation as you're writing the code. Don't wait a month later to write your documentation, because I guarantee you're going to forget how to use that particular function. So I always try to get people to make this their practice, right? Just to write documentation as they're coding. And then typically, so like I said, like I have these scripts kind of split up, but what I'll typically do as well in the projects, I'll have like a single run script. Basically what this run script will do is it'll call all the different functions I might need, that's starting from like the raw image file all the way to getting like a plot for a figure. I'll just have it in there, and that's like a way to kind of also document like how to use the script, right? So in the future, when I come back to this project and I might have forgotten all the details of the scripts, I can at least look at this main run script and figure out how to run it again. And then, this is a question that I typically get asked, like, how much data should you store? Obviously, you want to store your raw data, but, and maybe this is a little controversial, but I typically don't bother storing any kind of intermediate data. And what I mean by that is that any data that can be reproduced by running the code, like time series data, for example, I normally don't store that. But if there's any changes that are made to the data, so for example, if your code makes segmentation masks that you have to manually correct, I will obviously keep that corrected segmentation mask, right? And for me, again, like this is just a personal choice, but it's primarily just to kind of save some of the storage space. But I know some journals now require you to upload some of this intermediate data. I know we recently submitted an article to Nature, and they actually require us to submit all the raw data for the graphs as well. So maybe this might have to change it in the future. Okay, and this is really my last slide here in terms of sharing data. This is still kind of a little bit tricky, I think, but there are now a number of repositories out there, data repositories out there that are being created that you can upload your imaging data sets, your analysis scripts and things like that. Zenodo, I think, is maybe the most popular one out there. It is that one is free, but I think you're limited to about 50 gigabytes per repository. And then there's a couple others like the Harvard Dataverse and the Bioimage Archive as well. I'm not going to list all the different, all of these have like their own limitations and pricing structure. So I'm not going to list all of that. But if anybody's interested, Beth has in her bioimagingguide.org, she does have a page on this where she lists out a bunch of different sites that you can look at. Okay, sorry. So I know there was kind of a lot of information, but that is essentially my talk. We're going to have questions, I believe, at the end of the presentations. So I will now pass on the session over to our next speaker, Kristen.
Hi everyone, 1 second, just bear with me while I get this up the way I want it to look. Everyone can see the presentation, hopefully. So my name's Kristen. I am a bioinformatics scientist at the Van Andel Institute in the optical imaging core. And I'm probably going to be reiterating a lot of the things that Jen has already mentioned in his talk, but I did want to have a little bit more of a focus on how our stuff is still a little bit of a work in progress and how we are trying to actively implement data organization as well as storage, and take a little bit more of a overview type look on how Van Andel as a whole tries to store and organize its data for all cores. That was the wrong button to press. Sorry about that. There we go. So, there we go. So I want to take a little bit of a focus on reasons to be transparently organized. And some of those things are that we as researchers, we want to know where our data are stored. But the other people who are working with us, whether they're collaborators or other members in the lab, they also need to be able to easily find and know where data are stored. And using informative or descriptive folder and file names does help with minimizing that kind of guesswork to find and locate data. And figuring out a consistent data organization schema for your group or for a lab will kind of help keep that type of searching to a minimum. And we also want to use this kind of approach because it'll be less likely that we'll lose track of data or its corresponding analysis pipelines or data frames. And of course, we don't want our collaborators to be upset with us, hopefully. So being transparently organized will hopefully help you with that. There we go. Data storage at the Van Andel Institute works in an on-premise engineered network storage approach. And we also have a high performance computer cluster that integrates with and can read directly to our network storage. For each lab, they have at least 20 terabytes of storage. And for each lab group, we have what's called we refer to as a drop folder location that is accessible to all core staff that might be um, collecting data on the behalf of the lab, and we can then immediately transfer that data to the lab's network location. And each lab group also has its own network storage location that has at least 10 terabytes of storage provided. One of the things that our network storage has built into it is a lot of redundancy, and that is both on the storage side as far as our drives go, the hardware that integrates all of our drives, and we also use selective offsite AWS storage. So that way, in total, we really have at least 2.3 times redundancy built into the way we store all of our data. So if something goes down, we hopefully have a backup of it somewhere. We also are trying to very actively enforce compliance, and this is just as an institute as a whole, with data accessibility as well as sharing. And then we do utilize AWS for archival storage for inactive data to help us with our overall active network storage data management. And I spoke with our director of our network storage about just overall data lifecycle, and he had some input on how to really think about our data lifecycle as a whole. And one of the things that he impressed upon me was that everyone, regardless of if you're a lab or a core, really needs to have a data lifecycle plan for all steps of your data acquisition as well as analysis. And so if we start with whatever primary data we may be collecting off of your instruments, that is going to hopefully be moved into a secure with redundancy built in network or cloud storage service. And that would be considered our active data because we're trying to build out analyses. Maybe we're working it into a publication, and we'll have both our analyses as well as quantifications, the raw data, and any intermediate steps that might be involved in order to get our findings from that data. And that might be something that, is around 2 to 3 years, depending on how long the project is ongoing for and how long it takes maybe to get out that particular publication. Once we get to potential endpoints of analyses, things are published, maybe we're not actively working on that data anymore. We want to start thinking about how that data can be moved into an archive system or repository for long-term storage. And so this could incorporate things like our analysis endpoints, the raw data that we've collected, and then if we have intermediate steps that are very important for us to keep, maybe we include those. But potentially a lot of those things we might be able to delete because they would be able to be reproduced following the analysis pipelines that were built out previously. And despite everyone wanting to always be able to hold on to all of their data forever. Data storage is unfortunately a limited thing, and equipment will age out and become obsolete. So we do want to actively think about potential end of life for our data, and this could be things that really haven't been touched in almost a decade. The equipment in and of itself that maybe that data was collected on. It may be obsolete, and it might be actually better or easier to recollect that data on newer equipment to repeat those experiments if needed, rather than to try and use that old data to revitalize some type of analysis. So these are things that, again, our director of our HPC suggested to think about, and that's something that he's trying to integrate into Van Andel as a whole. Um, Shifting gears a little bit, thinking about how we as a core, optical imaging core, manage our imaging data. Our personal policy, similar to what Jen had mentioned in his presentation, is that ownership and responsibility of the data really is on the lab and the PI. What we do is that any data that we collect for a particular lab as a service, we transfer that immediately into the lab's drop folder in their network location as soon as the analysis is completed or the acquisition of the images is completed. And on our end, for our computers that are connected to our different instruments, we do delete data as needed to clear up space, but we try to give people a heads up before we do something like that in case they, if they're using it on their own, they make sure that they've gotten their data off of the instruments if they haven't yet. And just for full transparency, our folder organization NAMA schema is still something that we are actively working on to try and improve organization as a whole. The current structure, though, that we tend to use, and this applies to all labs within VAI, is that each lab will have its own folder location within their network. And then within each lab, we will have a VARIcore generated data folder nested within. And within that, we have multiple subfolders for each different core department that can deposit their data that they collect for that given lab. So for the OIC, one of the ways that we sometimes organize the data is that each member of the lab will have its own subfolder, and then we break up the data collected into subfolders of the actual images, and we'll have dates of collection, the instrument and that it was collected on, and a description of the data itself. And then if there's any image analyses conducted on, or that I carry out on this data, I will have a folder that has the ticket number associated, which I'll get into in a little bit, and a brief description of the project so that way people know what that particular folder is for. And because we are a service that offers both image acquisition as well as image analysis, and this is something that's done by two separate people. I handle, soon to be three when Jen joins us, I handle the image analysis steps of any kind of request, and then my counterpart Lorna, she handles any service imaging or microscope training that might be conducted. So the way that we keep track of all these different requests that might be coming towards us is using Jira, which is a project management software from Atlassian. And we built out our service portal so that way users and clients will know what kind of services we offer, and then they can submit a ticket that is very specific to their needs. In this case, I'm showing you an example of an image analysis request where they can fill out a brief description of what that project is, the lab that they belong to, any kind of chargeback number or activity number that we need, and then a description of what they're trying to analyze or what they're trying to image for their particular project. And once those tickets are submitted, we can then keep track of these things very easily as far as where they are in progress. And this has been a huge game changer for us because we do service almost 400 different scientists. So it can become a lot of stuff very quickly at times. And so being able to have these bins where we can very easily see what things need to be done, what projects we are actively working on, and what projects have already been completed really helps with that project management. And within each of these tickets, we can keep track of how many hours we've put into that particular project, notes that we need to keep track of, and communication with the particular client. Depending on licensing that you have for Jira, there are free versions and then there's paid versions of it. You can link Teams chats, which I use to communicate with people about their projects, along with a you can also link GitHub repos as well to specific requests. And you can build in some automation as well, where you can automate report time tracking, and then you can link different requests to each other. So if you have an image analysis request come in along with an image acquisition request, we can link these two requests together so that way we know that they belong to each other or are related to each other. I personally use the way that these tickets are requested as a way to help with the naming schema of the image analysis folders that are then going to contain everything for the image analysis request. So just a quick example for this project specifically, I use the ticket number that's assigned, so OIC-181, and then a very brief summary description of the title that the client has given to this particular request. So OIC 181 and mitochondrial analysis, and then all of the relevant subfolders or scripts that belong to that particular project will be nested into that folder. And the images that were collected for that particular project, they will remain in their own subfolder for the particular image collection that was done for that project. Right. And then I also use these ticket numbers and naming schema to keep track of my code using Git as well as the GitHub repository. And the way that I personally structure these things is that each analysis project has its own GitHub repository with that match naming schema. So I know exactly which sets of code and intermediate steps belong to a given project. And this is just a very brief example of that same project I showed before, that OIC 181 mitochondrial analysis, where I will have some subfolders containing models that I might be working on, the actual Jupyter notebook containing all of the code, any kind of other additional scripts that I might be needing to write out for either image processing or some other things I'm trying to test. And then also, I want to point out that for each project that I complete, I try to generate a summary report that I can then use to remind myself as well as to more easily communicate with the clients what I did for this particular project. And a quick example of the outline and template that I try to follow is that I have the name of the report up top, so I know what request it belongs to. I can record, to let them know how many hours it took to complete this particular project, and then a link to the GitHub repo if I'm turning this into a PDF to also provide to them. A little reminder on how to acknowledge our cores and publications, because that is something that we're trying to really get better about as a community at BAI. And then I have some sub-regions for portions of the report to fill out about the analysis for reference. And when I'm writing these things, I really try to keep people like my future self, as well as the clients and other scientists that might be joining the lab in the future, picking these projects up in the future in mind. So that way it's easy for them to follow along and really understand why we analyze the data this way and what the data frames might look like if they're needing to refer back to them. And just briefly, some areas I think that our group could improve upon, since this is still a work in progress, is trying to figure out a common naming schema for both service imaging as well as analysis requests, and potentially to change up maybe how we're doing our network folder structuring. So that way it's less dependent on the lab members, because they're not, especially if they're trainees, they're not always going to be there forever. And in addition to that, figuring out a really good consistent subfolder structure that would work more across projects generally. The way that I'm currently structuring them is fairly project dependent, depending on what I need to do for that given analysis request. And because of some of the NIH guidelines that are coming out, we're trying to figure out really reliable ways to share imaging data. And as we all know, given the different types of sizes this data can come in, that can be a bit of a challenge. And there's not a ton of repositories out there. So we're thinking about doing something like establishing an Amero server on site, or figuring out what might be a good, reliable repository for sharing upon publication. But in summary, hopefully I am just impressing upon everyone that we really just want to be as good of data stewards as possible. And so some ways we can do that is finding consistent organization schema that work for your particular group. Really using redundant, reliable storage for your raw data. We don't want to rely on just internal or external drives in our computers, or that you can plug into your computer. Those have a very high fail rate unless they are solid state drives. So we really want to use things that have a lot of built-in redundancy to protect that raw data. Having an active plan for your data lifecycle, so that way you can properly maintain active storage, is really essential. Those are engineered solutions, and they are very limited in their storage capacity. So knowing when to shift things over to archival storage, which is very cheap and in theory unlimited, is a really good practice to have in mind. And using transparent tools for project tracking and organization just helps with that communication with you and your collaborators. And using version controls, like Jen had mentioned as well, like Git, or repos like GitHub for code management, is very helpful. And notes are absolutely essential. And for me, I found that summary reports really do help place everything that was critical to an analysis in one place, and it makes it very easy to refer back to later on. And with that, that was my final slide. I will pass it on to Thomas.
Hi, I will try and be extremely brief. We've got like 5 minutes to the top of the hour. So I'm trying to think what slides to show, and most of them I'll skip at this point. Anyway, so I'm Thomas Bingo. I'm a co-director of research informatics at the Minnesota Supercomputing Institute. There are a few institutes on campus that come together to kind of make the whole scientific computing work. And in this
Thomas,
talk,
I don't mean to interrupt you, but you actually have till 1:30. This is 90 minutes, so you don't have to rush through it. You have more than half an hour.
oh, good, excellent. Well, then I won't run. That's good. Okay, so we've got so within the University of Minnesota, we've got a few institutes that are involved in this regarding this this talk. And so research computing kind of covers all of the things around computing for research. The Minnesota Supercomputing Institute is one of the institutes under that umbrella. And then the Office of Information Technology is involved, especially in the networking. So we don't handle any networking that's outside the Supercomputing Institute, and we kind of rely on the on the campus IT services for that. We've got data providers. Core facilities are, of course, places where data gets generated the most, and a lot of them high throughput and high content. And basically heavy data sets come from core facilities. Cloud partners, we only touch in passing here, but we do have like contracts and with I'd say the two of the main cloud providers, and for we use them mostly for, as Christine was saying before, we use them for backup. So at the Supercomputing Institute, we have about 408 CPU nodes in our current cluster. We've got a number of GPUs too that's been expanding over the years. GPU computing has become really something that the research community has been asking for and needed. And so we've got about 32 H100s, another 32 of the L40S, and of the previous generations, we've gotten a substantial amount of NVIDIA's A100s and A40s. A number of those are dedicated for interactive use. So most of them are kind of in the batch queue, but some of them are in the interactive GPU. So for graphical applications, or if you're testing things out in an interactive manner, those would be like an ideal place. Data storage, we got about 20 petabytes organized in 2 tiers. We got the high performance tier, which is for sustained computation. It sustains about 200 gigabytes per second, about 1.5 million input-output operations per second. The tier 2 storage is where meant for like data that is not like you wouldn't store like your database there, for example, but it's more like for it's actually a very good fit for imaging data. And so that's the destination for like lower, not hot storage, but kind of semi-hot storage. Consulting, we have about 35 staff dedicated to research informatics consulting, and we're 2 co-directors, myself and Christy Hensler. My colleague who takes care of all of the bioinformatics, and our group takes care of image analysis, neuroimaging, and some of the more classical scientific computing workflows like computational chemistry and material science and things like that. So in particular, we do offer like both image analysis and image processing, support in using the high performance clusters, and especially like with a focus on large scale data management. So that's where we kind of jump in. The University Imaging Centers, which is our imaging facility, they have already some staff who are trained in image analysis, and so they kind of take care of all of the image analysis that say, for example, Fiji or things that can be solved quickly and with not a substantial amount of compute. Anytime where that crosses a certain threshold, then we're brought in, and we can help them scale that up or devise some workflow that makes use of the cluster. We work on most varieties of fluorescence and bright field microscopy, electron microscopy, some ultrasound many years ago, some video analysis, and then micro-CT and micro-PET. Recently, there's been a lot of interest in spatial omics, and that's an interesting, we have an interesting, the fact that the research informatics now covers both like classical bioinformatics and image analysis makes us kind of very well suited to deal with spatial omics data, because it's kind of a combination of image analysis and of RNA transcripts or proteomics, whatever is most relevant for the technology. We do have Globus. That's been it's been especially useful for data transfer. So it does allow you for making the maximum use of your bandwidth. It's from the University of Chicago. It's a nonprofit, but you pay for the service. It allows a very good data sharing capabilities too. You can create data shared collections, and so that means that you can add people to. On the back end, you can have like either tier 1 or tier 2 storage on the supercomputer, and then that storage can be shared with any basically account that is supported by the Globus auth, which actually includes like even Google accounts. So it's very good for data sharing. And a lot of institutions support Globus, so transferring data among institutions, which is useful for like larger national collaborations. Has been proven very good for data transfer in that sense. But they do offer a host of other services which we've only touched on in our since we've got Globus, and we've got they do have support like distributed computing, so you can create workflows on a cluster or any computing resource that you have access to, and then run those computing resources from the Globus interface, which is interesting. It does support some automation, so especially for like ETL operations, you can get the data from the core facility, run some processing, and put it somewhere else. That's supported by Globus Flows. And it has a good number of access control capabilities for creating groups and accesses and permissions and all that. So I wanted to show a use case of some of these resources coming together, and in particular about large tissue cleared imaging, and in particular applied to the mouse brains. So that's to give a scale of what the size of a mouse is, and that's an image of one of the instruments that we have is the Calibre ID RSG4, which is a confocal microscope, in fact, but it has a couple of interesting features to it, where it's a ribbon scanning confocal, so it synchronizes the head with the movement of the stage, and it kind of acquires the data in ribbons. And the other is that it's got long working distance objective that allows you to really get the whole mouse brain in a short amount of time and with cellular resolution. So the typical resolutions that you would see would be like 1 microns X and Y, and a couple of microns in Z. This is the pipeline for that. We come in at the tail end. There's a lot of work going in to get these brains, get them cleared, and then imaged. And then the data analysis is where we come in, and we have over the years developed like a pipeline of data analysis that is tailored to this particular kind of imaging. So how does the data flow? These are two different buildings, but conceptually they come from the university imaging centers, and they're connected with a special campus network which has 20 gigabit connections into the data center of the Minnesota Supercomputing Institute, and it goes into a particular place which is dedicated for data delivery. There is the institute subsidizes the first year of storage in that space, and then investigators can move it into their own project space, and investigators are actually given up to 20 terabytes of space that they can ask for projects. So basically they would move it from the data delivery to their own space. We do perform the data analysis, and so that is tightly coupled to the cluster. We can run the image analysis pipelines on the cluster, and then get it back into the data delivery, or send it directly to the PI project space. If for example, we're collaborating with them on a particular project, we can just deliver it directly there. If the data doesn't need any kind of processing, you can just deliver it directly to the PI's project, and that's a common for some of the labs that have staff dedicated to image analysis, and so they basically they don't need our assistance. We do have an on-prem archival solution coming up in the upcoming months. And otherwise, we do have the option of offloading some of the data to like Amazon Deep Glacier, for example, or the Azure equivalent. So in terms of the analysis pipeline, this might be interesting in terms of like where the compute gets done and what kind of compute. So the first step is to stitch the ribbons together. The company does provide some of the code, but we've developed our own for particular purpose of getting the data organized in the manner that we want, and also kind of having a bit more control on how the stitching is done. So the result there is like a series of sections. Each one of these is about 20,000 by say 20,000 pixels. And it's still kind of noisy. The confocal is run at a speed that, you know, kind of balances that signal to noise ratio with speed. And so the first step is we go through denoising step. This is and in the next slide I'll show well, this is like a detail of the results of the denoising. But basically, we're using a trained noise model, which you can use from Fiji directly in the instant denoising plugin. But we've extracted basically the code that runs the model, and basically we're just using the trained weights from that, and it's written by custom Python scripts, so that we can run the parallel computation on each slice on the cluster. Initially, we tried to run the plugin directly, but it takes much longer, and also it's a bit clunky because you have to kind of open up a new copy of Fiji each time you do it, because of the fact that if you denoise multiple slides, they kind of get stuck. So anyway, so we've kind of took out the core and wrapped it in a Python script, and then we have that also allows us to run it on the clusters. The next step is cell identification. So most of the images we get have some sort of concept of a cell. It could be the soma of neurons. Well, most of the cases, if it's the brain, it would be like the soma of neurons. We've got some other kinds of cells also sometimes labeled. But oftentimes the question is identify these cellular bodies and get their XY coordinates. So that's the cell identification step. For that, we use the cell finder from BrainGlobe. That works in two steps, an identification and a classification step. They both use, especially the identification step uses a GPU accelerated routine, so we can also use some of the GPUs from the cluster. And sometimes we do need to fine tune especially the classifier. The identification step is you can think of it as a kind of a standard identification step, but like a spot detection, if you like, something like that. And then the classification decides which one of those spots is actually a cell or noise. And that is the piece that we often need to fine tune. Finally, we got the Atlas registration. We are using the Allen Brain Atlas. We also incorporated into the BrainGlobe. The BrainGlobe has a nice suite of software that allow you to manipulate and pull in many different atlases, and we use the Allen Brain Atlas. There's a couple that have updated annotations too, so we've used that also at times, but they all kind of hinge on that atlas. At times we have like portions of a rat brain also, that like a rat hemisphere. We can also do the registration to the rat brain atlas that can also be pulled in from BrainGlobe. And yeah, so that's okay, so this is the last slide I want to show as kind of the things that we're thinking about. So currently the visualization is done through Imaris, the company providing the well, I'd say most of the users are very comfortable using Imaris or can be trained in using Imaris. For these large data sets, it becomes a bit clunkier to work anywhere else than the university imaging centers, we do have a couple of instances of Imaris that run in kind of think of it as a cloud VM, and they can open up the cloud instance and use Imaris there. But the data transfer speeds are really the bottleneck here, and the experience is not super great. So we're kind of thinking of ways that we could give a more interactive experience to the users with technologies that are coming out now. In particular, the backbone of this is really the OME and GFF, the next generation file format by the OME consortium that uses ZAR, a particular way of storing arrays that is compatible with cloud storage, and in particular, like S3 is a very good example. So it breaks up the image in chunks. The chunks get stored as individual objects in the object storage. Think of it as a files in a directory. And they're also stored at multiple levels of resolution, similar to what Imaris and OME-TIFF and some of the digital pathology formats do. So they store different, like in a pyramid kind of fashion. You store the full resolution image, but you also store copies of the image in a lower resolution. So what I'm going to try here is I opened up this link here. So this is not an image we took. This comes from EBI, so EMBO essentially. But the bizarre bit is basically just the idea of what we're kind of expecting. And so you do get like basically an interactive way of visualizing the image, and you can go through like different slides and different sections and zoom in and out, and all of those chunks get loaded as you kind of browse in and browse out and zoom in, zoom out. Very similar to what you would get in other things like Google Maps, or the same as what happens in Imaris. So when you zoom in, higher resolution versions of that image get loaded in. So I think the nice thing about this approach is that you you do the conversion once, you store it on the S3 based storage, and then your browser is actually doing a lot of the work. So when you open the page, in this case it's using Vizar. It would just load the code of Vizar into your browser, and then the browser is pulling in the chunks as you kind of zoom in, zoom out, exactly as Google Maps would do, for example. So this allows you to not need to install anything on your local computer, but still be able to browse the data. I might try and click on the other link here. This we haven't tested before, so hopefully it'll work. Neurogancer is the other well-known browser for imaging data, and it does allow you to, this is the same exact data set, but using a different visualization software. This one allows like more of a 3D feel. Of course, I'm on my laptop, and I'm not on campus, so I get terrible connection, and also on VPN, so bandwidth is not the greatest. And so you can see some of the effects of that right here. But the idea is essentially that you don't need to install anything on your computer. You can browse the data set in 3D, and it does have like a volumetric viewer here too that will pop up in a few seconds. It takes a while. But in the meantime, I think I've reached the end of the talk. And so I think we can now address questions from any of the three speakers. But Rich, I'll give it back to you.
All right, thank you guys. I really appreciate those talks, and they were very interesting. Very interested in this visualization tool because yes, we have Amaris, but we don't have multiple copies of it, and this would be helpful. But I will open this up to anyone who has a question. You can, if you raise your hand, Ken will unmute you, and you can put it in the chat, either one, because we have about 15 minutes left before this ends. So does anyone have any questions for either any of the three panelists that spoke today? Ben. So go ahead, Ben.
All right, thank you. Thank you all for your great talks. That was very interesting. I really enjoyed seeing that. Thomas, I wanted to get back to something that you had mentioned, because I think that you had said that the data transfer speeds were ending up being the bottlenecks in your workflows. And I think on an earlier slide you said you're maybe getting at maybe 20 gigabits per second or so. I don't know if that's right, but what transfer speeds do you think would be necessary to, speed that out? What should people be aiming for? And I'm also curious what Kristen and John are currently transferring at, and if that's sufficient in their cases as well.
Yeah, thanks. No, I was very unclear. I apologize. The mentioning the data transfer speeds was mostly like to be able to visualize the data set remotely. And so that's basically the speed that goes from this laptop I'm using right now to the source of the data. And because I'm using like a my home connection, it basically means that it gets the worst speed. But data transfer speeds like that we get from the core facilities to the Supercomputing Institute are very well suited for the data sets. We haven't seen any issue on that side of things. The data sets we're talking about are about a terabyte in size, typically, depending on what resolution you get. And on if even on a gigabit connection, you get about 100 megabytes per second. So that gives a gigabyte every 10 seconds, and so it takes a few hours to get the data transfers. But with Gopher Science Network, which is that 20 gigabit connection, you can bring that down. And at that point, the bottleneck is typically like the data storage device itself. So you're not limited by the network connectivity. It's more like the actual support, the NAS that it used to acquire the data or something like that, the intermediate storage. Oh, but in terms of like requirements, certainly, if you're working with these large data sets, like multi-gigabit is kind of would be like the minimum. Anything below that makes it impractical, really, to move the data. But to the point of what Rich was saying is that if you do get a lot of the data in a position where, like on an S3-based system in a ZAR format, for example, then at that point you can actually on a laptop even access the pieces of that image that you're interested in looking at any given time. And at that point, you don't need a high speed connection. Even like, for example, a lot of the investigators really want to just open up the image and start looking at it. And to do that, you don't need that high speed connection. You can get that feedback really quickly, and then once they want to do analysis, if it's on every single pixel of the image, then at that point you need the high speed connections. But if it's like grabbing regions from the data, then that's very compatible with even like lower speeds. And the ZAR format allows you to do exactly that. You can extract cubes of information without needing to read the entire image.
Thank you.
Okay, other questions?
Oh, I could add on to that a little bit if Ben was interested in some other approaches as well.
Sure.
Yeah. Yes, please.
Yes. Yeah, so one of the tools that our HPC team came out with, I believe last year, is using On Demand. So that is a virtual web browser-based tool that allows users from any internet connection to connect to and use our HPC node resources. And we have quite a few built-in tools available, whether it's virtual desktops or instances of like RStudio, Jupyter Notebook, VS Code, those kinds of things. And so what we've done on our end is installed in an accessible manner a bunch of different open source image analysis tools, so that way users can access their data and visualize it or analyze it from wherever they might be with an internet connection. but utilizing the internal high-speed connections of the HPC to their network drive. And we found that that is has been really, really great for allowing faster analyses, better visualization of data, those kinds of things. But we don't have instruments that collect quite as large of data sets as what Thomas was mentioning yet. So most of our stuff is just like multi-gig per second connections, but we do have options for like 10 gig per second connections that we could build out if needed to transfer data from instruments to the network.
Great, thank you.
Go ahead.
John, are you doing anything different? Or
Well, so I think for us, we know we're kind of like limited to whatever the internet speeds are in the building. I don't quite remember all these numbers off the top of my head, but I don't think they're particularly fast, like maybe 50 megabytes a second or something like that. But I know that with the new light sheet microscope, they've been trying to install like a gigabit connection. But it's quite, it's quite challenging, right? Because they have to install a whole lot of new hardware. And so I think that was just going to go from the computer in the basement up to like their wherever their storage servers were. I think they just had that one connection set up. But yeah, so that's kind of what we had.
thank you.
Yeah, I just wanted to comment, Christine. That's absolutely correct. Good point of computing directly and visualizing remotely. We also use Open on Demand, and that's exactly how some of the like slide scanning workflows work. So the slides get delivered, and then Open on Demand, you can open QPath and basically use it from the browser. So yeah, excellent point.
So any other questions? Because I have one for Kirsten. So I will go while other people are thinking, because we only have a few minutes left. So we're about to set up a brand new file server, a massive file server. And how easy is it for you to get users to follow the policy of where they're supposed to store their data? Because this has been a nightmare traditionally for us.
Like a networked storage
Yeah,
system.
like a huge
Yeah.
file. Basically, we're going to keep all of the data.
Yeah, so that was something that the HPC team really kind of constructed for the institute at large. And we're at a fairly small institute, so I think it was rather easy for us to be able to establish something like that, where basically each lab is given like a chunk of data. They do pay for it at a very low cost, but they're given an allocation, and then the PIs can structure it however they see fit for their particular group. But they auto subnest a couple different locations, so that way some of the cores have places where they can deposit their data without having access to, like, say, the rest of the data or folders within each lab group's location. As far as implementing things like archival storage and like data lifecycle, that's something that we're still brainstorming on how to best implement it and how to get the labs really on board with how to think about their data in that way. I know that the HPC director, he has like a very like, if you're not using it anymore, just delete it, like he has that kind of approach to it. But that's not always how we can think about our data. So that's something that like we're still working on, and we're definitely improving, but we're getting more people to like really take advantage of the AWS archival approach for like very old data that they really are not actively working on but want to keep safe. And that's very cheap, and it's been working fairly well for a lot of people.
So do you use like ACLs to control access?
Sorry, I'm not like
access
that.
control list.
Yes, I think we do something along that line. Like each lab is pretty locked down as far as who has access to folder locations. Like random people in the institute can't just like go to another lab storage. So we keep things pretty safe that way.
All right, thanks.
Yeah.
Are any other questions? Because we're just about out of time here. If not, I'd like to thank the panelists and remind you that November's is going to be one day later in the week because of Veterans Day. And hopefully we will see you all there. And the registration for ABRF's annual meeting in Pittsburgh is sometime in October. It's coming up soon. And there you'll be able to also register for the AI workshop. So
October 21st, Rich.
October 21st. I was hoping he would chime in.
There we go.
All right, thanks. And remember, this has been recorded, and Ken will send a link to the recording in the next few days. So again, thank you all for attending and the panelists, and we will see you in November for the second part of the image processing series. Thanks, guys.